All right, folks, what we are going to talk about right now in this video is our aspects of chemical digestion. Yes, most of you are already in lab and saw this, but this will be a good refresher as the week progresses for you to continue studying. And then for those of you who weren't here, you can fill in the blanks in the checklist. So we're on page 46 of the checklist. And what we're going to do is we're going to run 3 experiments that involve the digestion of protein, carbohydrates, and fats. So without any further ado, here we go. What we're going to first do is, on page 46, is we're going to show you the process of the protein digestion. And so what we have, as we go down, we're going to have 2 test tubes. And in those test tubes, we're going to add equal amount of protein, as your checklist said. And the protein that we add is egg whites. So that is the protein source, egg whites. And then what we're going to do in one of the test tubes, we are going to add a small amount of what we call the pancreatin solution. Now, the importance of the pancreatin solution in this experiment is this is what contains the enzymes which speed up the rate of the digestion. Okay? And then so in one test tube, what we're going to do is we're going to grab egg whites. We're going to put the egg whites in there. And in one test tube, we are going to add just distilled water. And in the other test tube, we are going to add the pancreatin along with the egg whites. And so what we end up having here is we end up with a solution, our test tubes looking like so. So we can see the egg whites down at the bottom. We can see this is the one with the distilled water, and this is the one with the pancreatin. Don't worry about knowing the milliliters of how much we added. There's other important stuff that we want you to know. The big thing is we want you to know what shows a positive indication of a digestion of protein occurring versus which one doesn't. So this is what the test tubes would look like after we're done. And then what we would do is we'd put these test tubes in an incubator for approximately an hour and a half. And what they're saying, and if you read on number 4 on your checklist right there, at 37 degrees Celsius. Well, 37 degrees Celsius is actually 98.6 degrees Fahrenheit, which emulates body temperature. So we let that go for an hour and a half, and then what we do is we pull the test tubes out. And then if we're looking at letter B now in your checklist, so we're just going to read along here. After incubating, add enough of Biuret's agent, Biuret solution, and here's the Biuret solution, this bluish compound. What the Biuret solution is, this is an indicator for protein. So what we are going to do is we're going to add Biurets both to the test tube with the egg whites in the water, the egg whites in the pancreatin. And so if you continue reading on that, is we're going to have 2 possible color changes. We're either going to have a pink or a purple color change. Now what the pink indicates, the pink color change indicates that protein digestion has occurred. A purple color indicates that protein is present, which would make sense. Protein is present in both of the test tubes. So that's why we would get at least one that would be purple, but we also hope that digestion occurred. Again, that's why we added the pancreatin with the enzymes. So what we're going to do is we would add the Biuret's reagent to each of the test tubes, and then what ends up happening is this. And so what we have here, this test tube on the right, you can see the pinkish color. That's an indication that protein digestion has occurred. And in the test tube that has purple only in there, it's an indication that protein is present. So the purple also indicates that no protein digestion has occurred. So when we compare those back to back, you can see the difference between pink and purple. And so again, folks, which tube shows an indication that chemical digestion of protein has occurred? That would be test tube number 2, because that is what contained the pancreatin. And so then, what pancreatic enzymes are responsible for the beginning of the digestion of protein? There are 3 enzymes that are found in the pancreatin that are responsible for protein digestion. And the first one is trypsin, the next one is chymotrypsin, and the third one is carboxypeptidase. And not only are they found in this pancreas solution that we produced in the lab or made in the lab, they are also enzymes that are found within our own pancreas in the human body. And then so when we go to letter D, why did the contents of tube 1 turn purple? Well, it turned purple because protein is present in the fashion of the egg whites. And then letter E, considering the demonstration of protein digestion, which tube is the control tube? The control tube is always the tube that does not contain the pancreatin. So in this case, test tube number 1 would be the control, and the experimental tube is always the tube that contains pancreatin. That would be test tube number 2. Okay. So what we're going to show you now is the fat digestion experiment. And so again, we're still on page 46 of your lab checklist. And if we go down the list, what we're going to do is we're going to add a mixture that we have already been made up at lab. And if we read it, it contains both milk and a litmus solution. So when we look at this, here is that solution right here. Almost looks like a blueberry milk. This is the solution that we're going to use for the fat digestion demonstration. And we also, too, what we will do is we will take this and we will pour it into 2 test tubes. Okay, and again, do not worry about how many milliliters we use. We're not going to test you on that, folks, but we're going to add basically equal parts to 2 test tubes of this milk and litmus solution. And then in one of the test tubes, in fact, in test tube number 3, we're going to add the pancreatin. And again, why do we add the pancreatin? The pancreatin is what contains the enzymes that are needed to speed up digestion. So what we're going to do, put those tubes again in an incubator for about an hour and a half at 37 degrees Celsius. And then the end result is, this is what we end up producing right here. So what we can see, we get one test tube comes out pink and the other remains purple. So right here, the pink test tube, that would be test tube number 3, because that is what we added the pancreatin to. And what this illustrates, the pink color, that illustrates that fat digestion did occur. Then now the tube that remains purple shows us no fat digestion. And that would have been test tube number 4. And so we put those back, and then if we answer the bottom question, what is the purpose of using litmus in this experiment? Again, the litmus is what makes the milk change. The litmus is quite simply a pH indicator. It's just a pH indicator. Again, what do we use pH for? To test for acidity. All right, so now we go over to page 47 in your checklist. What is the purpose of using milk in this experiment? And the milk is used because the milk is the fat source. Just like the egg whites were the protein source, milk is the fat source. And then as we go down, what substances cause the change to occur in test tube number 3? Well, the end product of fat digestion would be the production of triglycerides. And what ends up are the fat in the milk, are triglycerides. And the color change is responsible due to the presence of fatty acids of the triglycerides. And then what, although not indicated by the color change in tube 3, what is another end product of fat digestion? And before you all yell that out right now, the answer is glycerol. And then letter G, what is the pancreatic enzyme responsible for the digestion of fat? That would be lipase. And then for letter H, the test tube that is the control tube, that would be test tube number 4. And then the experimental tube would be test tube number 3. And finally, we're going to come to the digestion of starch. So what we do, what we use is we use this solution that we made up in lab. This is the starch solution. And then what we would do, again, we would put equal amounts into 2 different test tubes. And then as you're reading underneath the preparation in your lab checklist, in a test tube 5, so these would be test tubes 5 and 6. In test tube 5 is where we would add the pancreatin. And in test tube 6, we just have the starch solution without pancreatin. And then so then what we're going to do, then again, we put that in the incubator for an hour and a half at 37 degrees and let it sit. After the hour and a half, we come back out and we pull those test tubes out. And in this case, what we would end up seeing is right here. That's what we end up seeing no matter what. So we can't tell if digestion occurred or not. So it's just going to be a plain empty, a plain clear tube until we have to do a few things to it. Now in both test tubes, what we are going to do is we are going to add Benedict solution. And again, you can see the Benedict solution is kind of that teal or aqua blue. We add that into both test tubes. And then what ends up happening is, what ends up happening is we would have 2 test tubes looking like this. But that's still not enough to tell us if carbohydrate digestion has taken place or not. So what we have to do is we'd have to take both test tubes and put that in a warm water bath, because what we need to do is we need to heat up the solution to where the Benedicts will react with the sugars that are in the solution. And what ends up happening is we would get 2 tubes that would look like that if we stuck them together in the water bath. One would remain blue, the other ends up coming out an orangish color. You can even say a red color, a green, a yellow. There will be some significant color change that occurs from after we put this into a warm water bath. Okay, so now as we come down, as we keep talking here, as we come down, what was the color change seen in the sample from tube 5? And again, tube 5 is the tube that we added the pancreatin to. So what you would see is you would see some color change. It would go from blue to a significant like red, green, orange, or yellow. And then what is the color change seen in tube 6? There is no color change. The tube remains blue. And then as we keep moving down, what color change seen in tube 5 indicates the presence of what specific carbohydrate? The answer is maltose. Now we would expect it to be glucose because that is truly the end product of carbohydrate digestion, but we do not let the experiment go all the way out to the very end. So what we do is we take the carbohydrates and we break them down from complex carbohydrates down into disaccharides. And the primary disaccharide that we break them down into is maltose. If we let the experiment go all the way out, it would eventually we convert it into glucose. But for right now, the answer is maltose, and that's what we want you to know. And then what pancreatic enzyme is responsible for the digestion of starch? That would be amylase. Again, yes, it's found in the the pancreatin solution, but amylase is also found in our human body, namely in our pancreas and also in our salivary glands. And then finally, why was there no color change expected for tube 6? We didn't add any pancreatin, so we did not have any enzymes. And then finally on page 48, folks, which tube is the control tube? That would be test tube number 6 because it contains no pancreatin. And the experimental tube is test tube number 5. Digestive experiments are done and complete.